Alright, cool. This is AQA A-level biology topic 2 completely patterned here. All of cells here. Gonna make it easy here. Alright. Light microscopes here. So light microscopes here, they're gonna use a pair of convex glass lenses here that resolve images to zero point 0.2 micrometers apart. So that's the resolution here for a light microscope here, 0.2 micrometers. Now, you might be thinking here, how is that useful to me? Why do I need to know it's zero point 0.2 micrometers here? But actually note this down, memorize this here. That's the resolution because in you need it in the essays here, and you also need it in the comparison questions as well. Trust me, they'll like that. They'll give you extra marks for that in things like essays, yeah. This is because the wavelength of light restricts the resolution. So the reason why the resolution is so small, yeah, if you can store this in your mind, is because obviously the wavelength of light is quite big, yeah, compared to like an electron. Light has quite a big wavelength, so therefore it's gonna restrict the resolution, yeah. And the definition for resolution is this third line here. That's resolution is the minimum distance between two objects at which they can be distinguished, yeah? So write that down here. That's the definition of resolution, yeah? The minimum distance, so how small of a distance you can see between two things to say those two things are separate. You get me? And the magnification of a microscope is how much that says bigger, yeah? Nothing else, yeah? An image appears compared to the original object, yeah? Don't even beg that, yeah? And the magnification is obviously the image size divided by the actual image, yeah, because obviously if something appears very small, yeah, in the thing, but it's actually big in the microscope, yeah, therefore it's going to be the image size, the size of it in the microscope, yeah, divided by the actual size of it. That's how many times it got magnified. You get me? So yeah, okay, cool. So how it works here is basically the light will pass through the condenser lens, yeah, which focuses and concentrates the light onto the specimen. So imagine you got a little specimen there, and you got the light coming through the condenser lens, yeah. That when it's coming through the condenser lens, it's gonna be like condensed onto the specimen. So you got bare light condensing, concentrating on that specimen, yeah. So it lights up the specimen. Obviously, they say that what you see is the image created through light. You know what I'm saying? Okay, cool. So therefore, the specimen, which is placed on a glass slide, interacts with the light passing through it. Yeah. So the specimen on the slide, obviously, there's bare light passing through that specimen. Yeah. So when it passes through that specimen, it's gonna interact with it. It's gonna, you know what I'm saying? The light's gonna be absorbed in different ways. It's gonna pass through in different ways. You get me? And depending on the properties of the specimen, such as its density, the thickness, it might absorb, reflect, or refract the light differently in different ways. And because of that, light from the specimen then enters the objective lens. Yeah. The objective lens is the primary magnifying lens of the microscope. So that's the lens that you actually see through first, the fixed one, before you go all those ones that are alternative that you can also increase the magnification. This is the basic lens. And that just magnifies the image of the specimen, yeah, and collects any of the light rays that are bouncing off the specimen from different points. You get me? So, yeah. They have a lower magnification than electron microscopes. You need to know that. And they also produce color images. Yeah. And you can also view living samples. So these are some advantages that they have color images and you can view living samples, yeah. Whereas some disadvantages, they got lower magnification, yeah, and they also got lower resolution. So this is what I was waffling about, bro. Is that you got the eyepiece there that you look into. You got the objective lens here. The light's gonna come there on the condenser shit. It's the light's what's gonna come through the condenser. It's onto that. It's gonna go through that, and it's gonna make different contour lines that your eyes will be able to see as the image in it, and that's gonna magnify it. And then it's gonna come through the objective lens, which is gonna magnify that, and then you see it there. Electron microscopes, yeah. So electron microscopes, they use a beam of electrons. So they're not going to use light anymore. They're going to use a beam of electrons, yeah, which is from an electron gun, yeah. And that's going to be focused by electromagnets, yeah. So electromagnets effectively do the same shit as a lens. You know how the lens makes it do what it does? Yeah, the electromagnets are going to make it do what it does. You get me? So yeah, they're going to concentrate the beam of electrons, yeah. They're going to focus the beam of electrons, yeah. I swear that's not how vacuum is spelled, but fuck knows, I repeated it as well. The vacuum environment is needed to that particles in the air do not deflect electrons out of the beam alignment, yeah? So the reason why you need a vacuum, yeah, is because obviously if there's other shit inside the environment, yeah, it's gonna obviously deflect the electrons. The electrons are gonna collide with them. It's gonna fuck up the whole situation that you got going on. So that's why you need it to be in a vacuum environment so that's just pure electrons that are moving. You get what I'm saying? Alright, cool. There's two types. First one is transition electron microscope, yeah. This is when a beam of electrons passes directly through a spin specimen, yeah. And the areas of that specimen that absorb electrons are going to be darker on the micrograph, yeah. So you're going to see them as darker, and the areas that the electrons went straight through, you're going to see as lighter. Do you see that? So transmission electron microscope, it's not transition, transmission, yeah, is when it goes directly through it, yeah. Alright, cool. Scanning electron microscope is when a beam of electrons passes along the surface, yeah. So a beam of electrons is a different type of electron microscope. You got TEM and SEM. Yeah, for SEM scanning, the electrons like scan across it like a computer. Yeah, it scans across the specimen, and it's gonna scatter obviously because that specimen has like bumps and has fine lines and that kind of thing. The electrons are gonna scatter across it and create different patterns. Yeah, and contour lines. Yeah, and that's gonna produce a 3D image. Do you see what I'm saying? Because obviously it has the different depth to it. Yeah. And yeah, you get me. Obviously, the reason why the resolution is very high for electron microscopes is because the wavelength of an electron is very small in it. Alright, cool. Because of the vacuum, yeah, live specimens cannot be observed. Because it's in a vacuum, obviously nothing can breathe in a vacuum. Therefore, obviously, my man can't breathe in a vacuum. So therefore, obviously, he's not alive, innit? And there's also complex staining processes, yeah, which might lead to artifacts. Yeah, artifacts is when there's other random shit inside the image that's not supposed to be there. Yeah, that's called an artifact because of the complex staining process that has to undergo. Also, yeah, the complex staining process. That's also a reason why the my man can't be alive. You get me? Okay, cool. Specimens, yeah, also need to be very, very thin so that the electrons can either scatter across them, yeah, or the electrons can, penetrate them. You get me? It has to be very thin in order for that to occur. SEM has a lower resolving power than TEM, so transmission has a way higher resolution since we're actually, penetrating it with electrons, whereas SEM is just scattering and that sort of thing. So that's going to be a lower resolving power, yeah. And also you got to remember they produce black and white images, so that's a disadvantage, yeah. Remember the other shit produced color images, yeah, whereas this produces black and white images, yeah. Alright, calm. So this is a pollen grain. So this is under SEM, as you see on the left hand side. So you see how it's 3D. It's black and white, but it's 3D, and it's obviously high resolution here. These are like virus and shit like that. And this on the right hand side is transmission. Yeah, so you can see it's a lot bigger detail. We're seeing inside the actual cell, all that shit, and that's obviously TEM. Okay, cool. Cell fractionation. Yeah. So this is basically a process. Let's say I've got a nucleus and shit. I want to separate that nucleus. I want to study the nucleus. I want to see it, yeah? We're going to have to break apart the cell and extract the nucleus, yeah? That's called cell fractionation. We're fractionating the cell. We're breaking up the cell into its constituent parts here. So this is done in two steps here. This is done in homogenization and ultracentrifugation, yeah? So I'm going to talk about homogenization first. So homogenization, yeah, it just means breaking up some shit. So the cells have to be broken up, obviously, to release the nucleus, to release the organelles that are inside of it, yeah? So to do that, you just basically just put it in a blender. So the cells are homogenized in a blender or in a mortar and pestle, yeah? That's the shit that your mum cooks with, yeah? And it's done in the cold isotonic buffered solution, yeah? So I'll explain why it has to be cold isotonic and buffered in a minute. But you just need to memorize that it needs to be a cold isotonic buffered solution. So normally they put like ice inside a blender and you'll cut it up in a blender like that. The solution is then filtered to remove any large debris here. So let me show you the situation now. So the reason why it's got to be a cold temperature here is because if you cool the solution, it's going to slow down the enzymatic reactions here. Because obviously when you're breaking apart the cell, all the enzymes are going to be released now. And those enzymes might fuck up the components. You get me? So when you put it in the ice here, it's going to slow down the enzymes reactions. So that therefore it's not going to degrade the cellular components. Do you see what I'm saying? Alright, cool. Buffered, yeah. The reason why my man's buffered, yeah, is because buffering the solution helps maintain a stable pH level, yeah. If you buffer the solution, the pH level is gonna be, sustained because any changes in pH, yeah, it could denature the proteins, yeah. Because like you don't know what environment you're cooking it in, right? You could blend this in some mad environments, yeah, and that's gonna cause the organelles to get denatured, the enzymes inside of there and stuff like that, or proteins to get denatured, yeah, because the pH has changed. So making sure that it's a stable pH level, buffer pH, prevents that, yeah. And then the reason why it's got to be isotonic here, isotonic means that the solution inside and outside is the same water potential, yeah, same osmotic pressure, yeah. So using isotonic solution helps to prevent damage to cellular membranes. If the solution was hypotonic, that means that there's lower osmotic pressure, there's less water in the cell, yeah? The water would rush into the cell and it would burst it. It would destroy the organelles, yeah? And on the other hand, if the solution were hypertonic, yeah? So there's a higher osmotic pressure, there's more water outside the cell, inside the cell, then water's gonna move out from the cell, innit? Water's gonna leave the cell and it's gonna cause the cell to shrink and then die. So obviously that's why it needs to be isotonic as well. Okay, let's talk about ultracentrifugation, yeah? So this is the second step of and the final step of cell fractionation, yeah. So what happens here is you get a tube of homogenate. So we call it a homogenate, yeah. That's the shit that you're gonna separate, yeah. So that's the shit that you homogenized, that you broke apart, Now it's looking all green, looking all crazy, yeah. That shit there, you're gonna put that into this machine over here called the centrifuge, yeah. And this basically just spins it bare fast, yeah. It just spins it fast, and what happens is because the shit's heavy, it moves to the bottom and it forms a pellet like that. So a tube of homogenate is placed in the centrifuge and spun at a low speed first, yeah? You need to remember that. Spun at a low speed, yeah? And the heaviest organelles, like the nucleus, yeah, is going to form to the bottom first. You need to remember this. The nucleus falls first, yeah? It's going to fall to the bottom, and then the fluid that remains is called the supernatant, yeah? So the shit that's blended, the nucleus and all that shit is going to fall to the bottom. It's going to be a pellet. And the shit above it, the liquid that remains, yeah, that contains all the other organelles, it's called the supernatant, yeah? Then what you do is you take out this supernatant, you take that, you transfer it into another tube, and then you spin it again at a higher speed. This higher speed is going to allow the second heaviest organelle, which is the mitochondria, to fall to the bottom. So you're going to get a pellet situation like this again, but that pellet is going to be mitochondria now. You get it? Then you label that mitochondria, label the other one nucleus here, and then you keep doing it again and again. The supernatant, you'll keep repeating it. You'll keep taking the supernatant out, put it in another tube, put it in the centrifuge at a higher speed, and then the next heaviest organelle is going to be left at the pellet. You get it? So that's how we're extracting the organelles. Then you can look at it under a microscope, that sort of thing. But that's how you actually take them apart. Yeah. Alright, calm. So here's a bit of a situation. So before it's just this supernatant shit. When it spins around there, it's gonna be collected at the bottom like that in a pellet. You see that? So that's called a centrifuge, yeah? Alright, calm. Alright, cool. I'm gonna talk about all the different parts of the cell now, yeah? So nucleus, yeah, has a double membrane structure, yeah? It has 3,000 nuclear pores, yeah, that allow molecules to enter and leave. So it has these nuclear pores, yeah, that allow obviously the molecules to enter and leave during, protein synthesis, during whatever, yeah. They contain a nucleolus, which is the smaller like nucleus inside of the nucleus. So here, this whole thing is a nucleus here, but inside of the nucleus, there's another nucleus called the nucleolus. That's what the actual, genetic information is, yeah. And then the nucleoplasm that surrounds that, yeah. And then the nuclear envelope is here. As you see, it's got pores around the whole situation. And then you've also got chromatin inside there, which is going to condense into chromosomes, yeah. So it contains a nucleus which produces ribosomes and a jelly material surrounding called the nucleoplasm, yeah? And it's a site of DNA replication and transcription and contains the genetic code, yeah? Alright, calm. That's enough said about that. Endoplasmic reticulum, yeah? So there's two types of endoplasmic reticulum, yeah? There's two types, yeah? There's rough and soft, yeah? Rough and smooth, yeah? So rough endoplasmic reticulum, yeah, is a series of sacs surrounded by a membrane with ribosomes on the surface. So the ribosomes are actually on the rough endoplasmic reticulum. As you see, this is the rough ER, yeah, and it's got ribosomes on that. Do you see the ribosomes there? There's a purple dots here. So and then the smooth ones there. So for the rough one, yeah, what they do is they basically just process proteins that are made on the ribosomes. So ribosomes are made on the proteins are made on the ribosomes, yeah. And then obviously there's protein structure, like it obviously gets folded and it obviously gets made into that 3D tertiary structure, that sort of thing, yeah. So the rough endoplasmic reticulum, it just processes the proteins. They move through here and it processes them, yeah. It processes them and folds them, yeah. And the smooth endoplasmic reticulum does the same shit but for lipids. So it produces, so it makes lipids here, and it also processes the lipids as well. Calm. And these are called cisternae, yeah. These like little folds here. Alright, cool. Golgi apparatus here. These process and package proteins and lipids, yeah? So they do the same thing as last time, but they package and process proteins and lipids. They do both, yeah? And they also produce lysosomes. That's a big part of them. You'll see what lysosomes are in a minute, yeah? Another thing they do as well is they modify proteins and they modify lipids. So they can produce glycoproteins, yeah, by adding carbohydrates to proteins, yeah? And that will make a glycoprotein, which can be used as like an identifier on certain cells. It's like a fingerprint kind of thing, yeah? Like if you have a glycoprotein, yeah, like the cell will be able to identify like what cell that is and that sort of shit. They're also fluid-filled sacs, yeah, with vesicles around surrounding the edges, yeah. So these are the vesicles around the edges. They're obviously fluid-filled, yeah. Okay, cool. Mitochondria, yeah. So they are oval shape and bound by a double membrane called the envelope, yeah. So that's the envelope here around the edge of it. You've got ATP synthase because obviously you need to make ATP. The inner membrane is folded to form projections called cisternae. So again, you've got these cisternae inside here in the inner membrane, yeah, these folds here. They also contain a matrix, which is has the enzymes for respiration. So you need to know that this inside here, this whole inside part is called the matrix, yeah, that blue part there. The matrix here is what allows the respiration, is what allows enzymes for respiration, yeah. So the matrix contains the enzymes. And it also contains mitochondrial DNA, yeah. So it actually does contain DNA and ribosomes, yeah. Um, One theory as to why the mitochondria and the chloroplast contain these DNA, yeah, is because some people believe that bacteria actually evolved into these, man, innit? So bacteria over many, many years evolved into mitochondria and they evolved into chloroplast or whatever, innit? So that's just some theory, yeah. Site of aerobic respiration, obviously, and ATP production, and also the DNA is used to code for the enzymes needed in respiration. So these enzymes need to be coded for, so that's why the DNA codes for them, yeah. That also explains, by the way, that evolution theory explains as well why they're very small in it, because prokaryotic cells like bacteria, they're the same size as mitochondria and chloroplast. Alright, cool. They also contain the same type of ribosome as well. I'll talk about that in a minute. Lysosomes, yeah. So these are vesicles that contain digestive enzymes. So lysosomes just have enzymes. They secrete enzymes. Yeah, they can hydrolyze phagocytes, release enzymes outside the cell. That's exocytosis, and destroy dead cells. Autolysis. Yeah. So autolysis is when the lysosomes use their enzymes to destroy dead cells inside, or they could use their like shit to hydrolyze other cells, such as like phagocytes. Yeah, which we'll see in a minute. Yeah. We're talking about like immune response and that sort of thing. You'll see it in a minute. But effectively, what they do is they contain digestive enzymes and they release them. So that's where enzymes come from, is the lysosomes, yeah? And as you see, this is mitochondria. That's the nucleus there. Lysosomes chilling over there. Okay, cool. Ribosomes, yeah. Ribosomes are composed of ribosomal RNA or rRNA and proteins, yeah. They are made up of two subunits. So there's a large subunit and there's a small subunit. You need to know about these, yeah. All you need to know about them is that eukaryotic cells, yeah, contain large ATS ribosomes, yeah. And prokaryotic cells, as well as mitochondria and chloroplast, they have the small 70S, yeah. So AES is the large ones, yeah. 70S is the small one, yeah. And eukaryotic have the large, prokaryotic have the small, yeah, and it's the site of protein synthesis, yeah. So the ribosomes are on top of the rough endoplasmic reticulum, yeah, and they just produce protein, yeah. That's where proteins are made. Okay, cool. Chloroplasts here. Chloroplasts are composed of a fluid-filled stroma. So this whole thing is called the stroma inside, yeah, as you see inside here, and you got these thylakoids here, and when these thylakoids stack up, so that's one of these like circular shit here, and when those thylakoids stack up, yeah, we call them the granum. As you see there, the granum, yeah, is all of that stacked together, whereas one of these is just the thylakoid, yeah. Safe. And obviously, the stroma contains enzymes for photosynthesis, yeah. And the thylakoids, which stack to make granum, yeah, these are filled with the chlorophyll pigment, yeah. It contains the actual chlorophyll, yeah, which is obviously what makes it green and what gives it certain things, yeah. And chloroplast is surrounded by, and obviously lets it do photosynthesis, yeah, absorbs light or whatever, yeah. Chloroplast is surrounded by a double membrane as well, and it's also obviously the site of photosynthesis, yeah. Okay, cool. Vacuoles, yeah. So vacuoles. So these are the fluid-filled sac that you find in plant cells, and the smaller one in human cells as well. So the vacuole is a membrane-bound organelle found in the cells of plants, fungi, protists, and some animal cells. Yeah, it is essentially a large fluid sac within the cell's cytoplasm. Yeah. Vacuoles serve as storage components. So this is what they do. They just storage. Yeah, they just include like water, ions, nutrients, sugars, proteins, and that kind of thing as well. Waste products as well. Yeah. And so basically, you need to know as well that the accumulation of water within the vacuole, it creates that turgor pressure, yeah? The turgor pressure, it helps maintain the rigidity and the shape of the plant cells, yeah? So this pressure is crucial for supporting the plant structure and facilitating processes such as the cell expansion and growth, yeah? So what I'm trying to say here is that the water inside the vacuole creates this turgor pressure. You need to know that word, turgor pressure, yeah? And that basically helps the rigidity of the plant cell and keeps it in a straight line, keeps it in a compact sort of way. This extra shit you don't really need to know, but you can know it. It might help you in the future with the essays and shit like that. Some vacuoles contain pigments such as cyanins, and betalains, yeah, which give the plants their colorful hues. That's why some plants have nice little cool colors and all that. Some flowers, whatever. It's because of the vacuole that actually contains those colorful hues, yeah, because of these kind of chemicals, yeah. And what you also do need to know is the word tonoplast as well. The tonoplast is the membrane that surrounds the vacuole, yeah, in plant cells, yeah. So as you see, this tonoplast there, that's the membrane around the vacuole, yeah. Prokaryotic cells. So this is shit like bacteria, yeah. These are much smaller than eukaryotic cells, yeah, and have no membrane-bound organelles. So the two things you need to know about prokaryotic cells, the two main things, yeah, is you need to remember no membrane-bound organelles. That's a very, very strong definition, yeah. And second thing is obviously no nucleus as well. So some have a cell wall as well made of murine, yeah. Some people call it peptide glycan or whatever, yeah, but it's just murine, yeah. Let's keep it a buck. And also it has no nucleus, obviously. and they have a capsule as well, which helps the cell retain moisture because they could dry out. So you need to know that has this capsule around it that helps retain moisture. That's one of the uses of it, yeah. And it also allows it to adhere to surfaces, so it lets it like glide along surfaces and stick onto surfaces and that kind of shit. The plasmid is a circular DNA. So the circular DNA that you see, you don't really see it here, but it's like small rings of DNA. That's called plasmid. And it also just has the normal like nucleoid, like the DNA that's just chilling on its ones, yeah. Flagellum, yeah, is a tail structure that rotates to move the cell. So this shit moves around to move the cell. And it's also got pili, which you can't really see here, but it's like hairs around the thing to, attach to other bacterial cells and move around. And instead of, lysosomes, it's got mesosomes. Mesosomes contain enzymes as well, and that's required for the respiration since these are obviously living cells, yeah. Viruses are obviously non-living, yeah, and they're acellular, which means they're made of just one shit. It's just one cell, and they're obviously non-living, yeah. They don't actually respirate and breathe and shit. They rely on other shit in order to continue. They consist of nucleic acid, so like RNA, and are enclosed in a protective protein coat known as a capsid, which can also be covered by a lipid layer called the envelope here. So, like for example, here you got influenza. You also can see, HIV, what it will look like in the future. You just got the RNA inside there. You just got an envelope around it, and you got like a little capsid protein coat inside, and that's really it. And it's just got spikes and stuff like that, so that it can attach to receptors and that sort of shit. Same as this bacteriophage bullshit. Same thing. It's got this little head here. It's got DNA stuck in there. It's got a neck tail. It's got things to move around. Okay, cool. The cell cycle, yeah. So this is easy. Yeah? This don't trip over this. Yeah, a lot of people find this difficult for some reason. I don't know why. Yeah, there's three stages to the cell cycle. Yeah, interphase, mitosis, and cytokinesis. Okay, cell division is mitosis and cytokinesis. Interphase is like the preparation phase, okay? Now, for interphase, we've got three different parts to interphase. We've got G1, we've got S, and we've got G2. G1 is mainly cell growth, S is the DNA replication, and G2 is going to be preparation for mitosis. So let's talk about interphase first. So interphase is the longest stage of cell cycle and contains three subphases, yeah? G1, which stands for gap 1, S, which stands for synthesis, yeah, because your DNA replication, and you're doing G2, gap 2. So in the G1 phase, the cell grows and synthesizes proteins and prepares for DNA replication. In S phase, so that could be proteins such as enzymes, yeah. In the S phase, DNA replication occurs. Obviously, we know about semi-conservative replication from topic 1, yeah. So obviously that's what's going to happen. DNA replication is going to occur, yeah. And the cell creates an identical copy of DNA. The reason why is because you're going to produce two cells in the end, yeah. So therefore, you need to double your DNA so that it can split and produce two cells, yeah. And then the G2 phase there consists of further cell growth and preparation for the cell division. And the cell also produces any additional organelles and proteins that are needed for mitosis, yeah? So let's talk about mitosis now. So this is the second stage, yeah? So the interphase is just preparing it, making sure the DNA is ready, making sure the cell's ready. Now we get into the real shit. So mitosis has one round of division and is used for growth and repair. The cells are diploid and genetically identical. So the cells are diploid that are produced, and they're also genetically identical as well that are produced, yeah? So this is a little overview. So obviously, in order to build shit, you need the cells to replicate, so you have more cells to build shit with. So obviously, interphase occurs that you got DNA replication. Prophase will happen, which we'll see in a minute, where it condenses the shit inside there. Is going to condense into actual chromosomes. The chromosomes are going to meet in the middle. That's metaphase. They're going to move away. Anaphase is going to split the nucleus inside of that, which is telophase. And then cytokinesis is the final step, which is technically not included in mitosis here. Hence why it hasn't stated a name for it here. Okay, cool. Prophase. So this is the first step here. So the main things you need to know about prophase is the nucleolus disappears and chromatin condenses into chromosomes. That's an important thing. Chromatin condenses into chromosomes. Also, the centrioles separate and spindle fibers form. So these are centrioles, yeah, and this is where the spindle fiber comes from, which is gonna attach to the fucking what do you call it? It's gonna attach to the chromosome, yeah. So these spindle fibers they start forming from the centrioles, yeah, and the centrioles will split and they'll go to opposite sides. The chromatin, so the DNA that was just chilling there, is going to condense into chromosomes, and the nucleolus is also just going to disappear. So this is all happening just in the nucleus alone, yeah? Okay, cool. Metaphase. Now what's going to happen is during metaphase, the chromosomes are going to align across the metaphase plane, yeah? Or in other words, the chromosomes are going to link up in the middle of the center of the situation, yeah? and it makes sure that's equidistant between the two spindle poles. What that means is just equal distance is directly in the middle, so it's equal distance between two spindle fibers, yeah. Otherwise, between two centrioles, yeah. Otherwise, you could get some mad situations going on where like there's too many chromosomes on one side, yeah. Each chromosome is attached to spindle fibers originating from opposite folds at the kinetochore, yeah. Alright, say nothing, bro. Yeah, that's a bit of overkill there. Cool. The alignment of chromosomes at the metaphase plate ensures that each daughter cell will receive identical set of chromosomes. So all you need to know here, bro, is that it attaches to the spindle fibers, yeah. The centrioles, it meets in the middle, and that's really it, bro. Middle for metaphase, you get me? That's all, bro. Anaphase. So anaphase begins with separation of sister chromatids. So once the chromosomes have been separated, and as you see, the thing that holds them together in the middle is called the centromere. So the centromere is inside is the holds together two chromatids to form one chromosome, yeah. And these chromosomes get broken apart to make chromatids, yeah. And these chromatids will obviously move towards each side. They call them poles, yeah. So anaphase begins. Ana means away, yeah. Ana, like anaphase, you get me? So the centromeres split. So the shit that was holding them together, this circle thing is gonna split, and the chromatids will be pulled away from each other, yeah, to the opposite poles of the cell. And at the end of anaphase, each pole has a complete set of chromosomes, ensuring that each daughter cell will receive a full complement of genetic material. You're probably asking, yeah, how does that make sense? You just split them. So how can it be? How can both of them have a full set of genetic material? You're forgetting that in interphase, we doubled them. So we had a certain set, we doubled that set, then we split that set. So we got two original sets, you get me? So that's why. Okay, cool. Telophase, yeah. This is the final stage of mitosis. So telophase marks the end of chromosome movement. Chromosomes reach the poles and begin to decondense. So they do condense back into chromatin, yeah, as these are going to be two new cells, so it's going to go back to its original form, yeah. A new nuclear envelope forms around each set of chromosomes and the nucleoli reappear. So what's going to happen is a new nuclear thing is going to go around them. So you see this nucleus there? The nucleus is basically going to split. So the nucleus is going to split to make two different nucleuses, even though we're still in the same cell. So we're in one cell, but we've got two nucleuses now. And then cytokinesis is when the cytoplasm is going to split. So the actual cell is going to split into two. So we had these two nucleuses that were produced a minute ago. And now cytokinesis completes the process of cell division, yeah, by dividing the cytoplasm between the two daughter cells. In animal cells, a contractile ring composed of actin and myosin filaments forms at the cell's equator and contracts. You'll learn more about actin and myosin in, I think, year 2, yeah. Pinching the cell membrane inward until the cell is divided into two. So this basically actin and myosin come and form a contractile ring, yeah, which basically pushes it together and it basically breaks the cytoplasm, it breaks the cell membrane, yeah. So then it splits into two new cells that were identical. Each one is identical to the original cell before it got duplicated, yeah. In plant cells, a structure called the cell plate forms between the daughter nuclei and develops into a new cell wall, yeah, dividing the cell into two separate compartments. So obviously, you also got the cell wall in plant cells. So that's what it's saying here is that the cell plate forms between the daughter nuclei and develops into a new cell wall for both of them, yeah. So if you want a little recap here, well, basically, interphase, we just replicated things, then we went through mitosis, and now we're just splitting it with cytokinesis, yeah. We're splitting the cell in the end. Binary fission, yeah. So this is the way that bacteria and prokaryotic cells replicate. Obviously, they don't have nucleuses in it. So what happens is just one cell, the circular DNA. So instead of the DNA replicating, the circular DNA, like the plasmids and shit, they replicate and attach to the cell membrane. And then the cell membrane grows in between the two DNA molecules and begins to pinch inwards, dividing the cytoplasm. So just jump straight to cytokinesis, no chromosomes involved, no nothing. And a new cell wall forms between the two, and hence two identical are made. So literally just within the same cell, the DNA just duplicates. It just splits, and that's it. It's simple, yeah. And this is the meiotic index here. So if it asks you to calculate the meiotic index here, it's the number of cells in mitosis divided by the total number of cells times 100. So by mitosis, I mean here you can tell that cell is clearly in mitosis here because the shit has condensed. They've condensed into chromosomes, that type of shit. So that's prophase. You see some are in anaphase, some are in that. And you take every single one that's in mitosis, you take the number of that and you divide it by the total number of cells that they give you in the image, and you times that by 100, yeah, and that's gonna give you that's called it because it's an index. That's why it times it by 100, yeah, and that's called the mitotic index, yeah. So yeah, cool, bro. Let's talk about cell membranes here. So all cells and organelles are surrounded by a partially permeable membrane composed of a sea of phospholipids with a protein in between here. The main function of the membrane is obviously controlling the movement of substances in and out of the cell. and obviously the fluidity and the way that's arranged there gives it a name of fluid mosaic model, yeah. So this is called a fluid mosaic model. The reason for that is it's like a little mosaic, you get me? You know when you got the mosaic tiles, all of them things, yeah. That's the situation that's going on here. You got bare different shit. Um, like in year 3 when they ask you to draw the mosaic thing, you get me? And it's got a sea of phospholipids, yeah, obviously. And the phospholipids, the way they work here is the head is obviously hydrophilic, yeah, so therefore it's attracted to the water because the water is going to be around the cell in it. Whereas inside the cell, it's going to be hydrophobic. So you see how the tails they position themselves towards each other where my mouse is, and their heads are away from it. That's because the heads are away, and because hydrophilic, and the tails are together because they're hydrophobic, yeah. Uh, Look, also the glycoproteins and the glycolipids outside, as you see, they stick out. Yeah, those are obviously used as those can be used for like anything. They're mainly used for they're used for different shit in it. They're used for like recognition, cell recognition. Yeah, so like to recognize a cell, they use as effectively like receptors. Yeah, and the cholesterol as well obviously holds the thing in shape in it as for structural support and that. Alright, cool. And obviously got different types of proteins as well, like channel proteins and the carrier proteins, yeah. Okay, cool. So the hydrophilic heads point outwards, yeah, and the hydrophobic tails point inwards, yeah. This allows lipid-soluble molecules to pass through the membrane, but not water-soluble molecules, yeah? Because the inside is hydrophobic, that means that lipid-soluble molecules can pass through the inside, yeah? But obviously water-soluble can't because hydrophobic. The peripheral proteins, yeah, provide mechanical support and integral proteins. So those are ones like carrier proteins and channel proteins. Those are ones that are integrated in the thing, so they act as like a pathway, yeah? Those are obviously those are obviously involved in transport, yeah. Whereas the peripheral ones, like the globular ones and the glycoproteins, those are just mechanical things. You get me? The peripheral proteins can also make glycoproteins or glycolipids, which are used as cell recognition. Yeah. Okay, so let's talk about channel proteins. Yeah. So these channel proteins, they're basically filled with water. Yeah. So they're just tubes that are filled with water. And as I said before, yeah, small non-polar lipid soluble molecules, they can just cut straight through the membrane. They don't need a protein or anything, yeah, because it's lipid soluble, yeah. But water soluble things, yeah, or polar molecules, yeah, they can't do that because they can't dissolve in protein, in lipid, you get me? So they gotta cut through here. So this thing is filled with water, so because they're water soluble, like ions and shit like that, they can just dissolve into the water and just move through. Okay, so channel proteins are tubes filled with water that allow water soluble ions to diffuse. Carrier proteins bind with large, yeah, so that's called facilitated diffusion, yeah. Normal diffusion would just be moving across the barrier, yeah, like the lipid soluble ones. But this is facilitated because it needed the channel protein to do it, yeah. And then carrier proteins, yeah, these are things like active transport and shit. Carrier proteins bind with large molecules like glucose or amino acids and will change shape to pump the molecule onto the other side. So it will change shape, it will pump it to the other side, obviously using energy, using ATP. Cholesterol makes the membrane more rigid, yeah, and reduces the lateral movement of phospholipids as well as prevents the leakage of water and dissolved ions. So yeah, that's what cholesterol does. Main thing here is it prevents the leakage of water and dissolved ions, yeah, from the cell surface membrane, and also it prevents lateral movement of the phospholipids, keeps them in place, keeps them tight. You get me? Simple diffusion, yeah. So this is simple diffusion. The passive movement of small non-polar lipid-soluble molecules like carbon dioxide or oxygen from an area of high concentration to an area of low concentration. So write that down. That's simple diffusion. So simple diffusion is the passive movement, yeah. So there's no active involved here, no ATP involved here, of small non-polar lipid-soluble molecules. It has to be lipid-soluble molecules, yeah, that are moving from my man to my man. You get me? Through the bilayer without any proteins, yeah. This happens directly through the bilayer. Facilitated diffusion, yeah. This occurs when the channel protein to transport polar charged and water soluble molecules, yeah. So as you see, the proteins here, they come through there, you get me? They come through there as a channel protein, you get me? Because the channel protein, they dissolve in the water in that tube, and that it obviously moves them to the other side. Osmosis, yeah. So osmosis is the diffusion of water molecules from an area of high concentration, so a very dilute environment, they call it. Dilute environment means there's bare water, to an area of a low concentration, which is concentrated technically. If it's low concentration of water, it means it's concentrated in other shit, you get me? Through a semi-permeable membrane. You need to say through a semi-permeable membrane, yeah? You can't just say movement of that to that, you get me? A-level definition, the more preferred definition, as you say, the movement of water from an area of higher water potential to an area of lower water potential through a semi-permeable membrane, yeah? Okay. Active transport, yeah? So this is the transport of molecules from an area of low concentration to an area of high concentration for a carrier protein, yeah? This obviously requires energy in the form of ATP. So the carrier protein will get my man from there. As you see, there's a low concentration there. Because it can't just diffuse across, it's going to change shape using ATP. ATP is going to break into ADP and PI, and that's going to release energy, which will cause it to change its conformational shape. And that's going to move that to the other side and pump it across here through the carrier protein. So that's obviously active transport. Okay, cool. So here's the actual process for active transport, so you can write it down so you can be, you know what I'm saying, crisp with it. ATP is hydrolyzed, broken down into ADP, adenine diphosphate, and inorganic phosphate by ATPase enzymes, obviously ATP hydrolase, yeah? Releasing energy in the process. This energy is used to power the active transport process. 2. The energy released from ATP hydrolysis induces a conformational change in the carrier protein embedded in the cell membrane. This change allows the carrier protein to bind to the specific molecule or ion to be transported. Yeah? The carrier protein undergoes another conformational change driven by the energy from ATP hydrolysis that moves the bound molecule. So first it changes shape, yeah. Then now that it's hooked onto the molecule, it's going to do another change here, which is going to push it to the other side against its concentration gradient from an area of lower concentration to an area of higher concentration. 4. Once the molecule or ion is transported across the membrane, the carrier protein undergoes another conformational change that releases the molecules. Now it's going to release it into the cell or the intercellular compartment. Yeah, so just write that down. And obviously, ATP can be reformed as well. Alright, cool. Cold transport. Yeah, so this is a bit of a new one. You need to know about cold transport. It's very important. You'll see it in the future. Effectively, what happens here is there's sodium ions inside this epithelial cell, yeah? And these sodium ions, through active transport, they get pumped out through this sodium potassium pump, and they get pumped into the capillary, yeah? So the whole aim of this, let's just talk about the aim. The aim is to move the glucose from, this is the lumen, the black area is the lumen of a small intestine. This is the epithelial cell in the small intestine, and this is the blood. So the whole point of this is to move glucose from the small intestine all the way across the epithelial cell into the blood, yeah? So the way, obviously it can't do that on its own. So the way that this works here is the sodium that's inside my man here, inside the epithelial cell here, it gets actively transported through this pump over there, through the sodium potassium pump here. And obviously that requires ATP for energy because active transport here. So ATP will break into ADP and PI. So it will move through there into the blood. So now you got increased sodium in the blood, yeah? Calm. Because of that, that's gonna lower the potential gradient. That's gonna lower the what do you call it? That's gonna lower the concentration, yeah, of sodium inside the epithelial cell. So now there's a lower concentration of sodium in the epithelial cell compared to up here, compared to the small intestine lumen, right? So now sodium just moves normally through facilitated diffusion into the epithelial cell since there was a lower concentration there, yeah. And as it does that, it moves through a co-transporter protein, yeah. And this co-transporter protein, yeah, it moves glucose with it. So it will basically like attach to glucose, it will come with glucose and they'll both move through it. So glucose and sodium will move through the co-transporter protein via facilitated diffusion as normal into the epithelial cell. So that's going to bring glucose with it. And then glucose, because there's a high concentration in the epithelial cell, will just move normally through diffusion into the capillary. So write that down bit by bit. So sodium and potassium ions are pumped out of the epithelial cell into the blood, leaving a lower concentration in the epithelial cell. Naturally, that creates a lower concentration gradient of low concentration of sodium ions inside the cell compared to the lumen up there. So therefore, sodium is going to move from the lumen into the epithelial cell. So sodium ions then diffuse through the co-transporter protein from the lumen into the epithelial cell down the concentration gradient. Hence, glucose or amino acids also attach to the transporter protein and move into the epithelial cell with the sodium. That's why it's called co-transport. You get me? And then since there's now a lot of glucose in the epithelial cell, it can naturally just move by facilitated diffusion into the capillary. You get me? So that's how it works, yeah? Okay, cool. Bacteria and viruses. Bacteria are prokaryotic and can survive on their own. However, viruses are non-living and hence they need a host to survive. Viruses are also a lot smaller than bacteria. Physical barriers to infection. So there's a couple physical barriers here. So it's not all about immune response here. There's a couple physical situations as well. So that includes stuff like stomach acid, yeah, that kills bacteria. Skin, which is obviously a tough barrier. It consists of keratin, yeah. Again, you can note that for the future. And obviously gut and skin flora that compete with the pathogens for food. So in the gut, yeah, and the skin, you got this flora, this healthy bacteria that people chat about, yeah. And that bacteria there competes with pathogens obviously for food and space. So that can also lead to the elimination of pathogens in it. Alright, cool. Non-specific immune response, yeah. So these are non-specific. These will just do it to anything. Doesn't matter what it is. So these don't involve antigens. So these are things like interferons. So specific would involve a specific antigen, yeah, that has come from a certain virus or certain whatever, and it's gonna specifically attack that. Non-specific means it can do it to anybody. So we got interferons here. These prevent viruses spreading by stopping protein synthesis in the viruses. So these interferons, they'll stop protein synthesis inside the virus, which will obviously stop it from spreading. Lysozymes, yeah. So these are enzymes found in tears and mucus. So you know your tear comes a bit, moves a bit salty, yeah, it moves a bit mad, and the mucus as well. Obviously, those are the lysozymes in there as well, which obviously kill bacteria and they damage the cell wall. Yeah, that's why you start tearing up bears when you get sick. Inflammation. When white blood cells are damaged, they release in histamines, yeah, which cause a vasodilation. So basically, how this works, yeah, I'll break this down for you. When white blood cells are damaged, yeah, they start releasing these histamines, yeah, which cause vasodilation. What that means is it increases the flow of blood. So dilation means it's gonna increase. So it increases like the bandwidth, yeah, of your like arteries and shit. So that's what's gonna happen, yeah, and that's gonna increase flow of blood to the infected area, yeah, and that increases permeability of blood vessels. As a result, yeah, the antibodies and the white blood cells and plasma they leak out into the tissue that's infected and destroy the pathogen. So because that tube, yeah, has dilated, it's increased, yeah, there's more blood around it inside that tissue, and so that causes some of the shit inside the blood, like the antibodies, the white blood cells, and the plasma, to start leaking out into the tissue around that, which is going to destroy the pathogen. You get me? So that's obviously what it also does. That's called inflammation, yeah. Phagocytosis, yeah. So let's talk about phagocytosis. So a phagocyte is attracted to a pathogen, yeah, by chemical products of the pathogen. So the pathogen's removing their mad things, yeah, like your girl, and obviously the phagocyte sees that and he starts getting attracted to that, yeah. He likes that. He likes what's leaking out of there. You get me? So he starts moving towards that, yeah. A longer concentration gradient, of course. Now, what the phagocyte is gonna do is it's gonna attach to the receptors on the pathogen, yeah. So the receptors on the phagocyte attach to the chemicals on the pathogen, so the antigen effectively. So it attaches to those. The pathogen is obviously anything that can cause destruction, like bacteria, virus, whatever. So the phagocyte attaches to those, and now what happens is lysozymes within the pathogen migrate towards the phagosome. So these lysosome, yeah, which has lysozyme, which is the enzyme that will destroy things, migrates towards where the pathogen has been engulfed. So this pink thing is the pathogen, yeah. So the lysozymes are moving towards that. Lysosomes are moving towards that, so that they can attack it with lysozyme and destroy it. And once it engulfs that ring there, that thing there is called a phagosome. You understand? So now that we got that phagosome, obviously the lysozymes inside of there are gonna break out into the phagosome and they're gonna dissolve the pathogen. So it's going to hydrolyze the pathogen, in other words. So the lysosomes release lysozymes into the phagosome where they hydrolyze the pathogen. So it's going to hydrolyze the pathogen, and the pathogen is going to break down into its molecules, into its antigens, yeah? And so now what happens here is the products of hydrolysis are absorbed by the phagocyte. So this shit that's been left over, it gets absorbed by the actual full phagocyte here. So the phagosome is that circle there. That's where the engulfed pathogen was here. And then the products of that hydrolysis, so the remains, the body, yeah, the remains of the pathogen, yeah, get absorbed by the fucking by my man, by the phagocyte, yeah, and the phagocyte actually presents them on its surface. So it takes those antigens, yeah, and it presents them on the surface of the phagocyte. So that then you call that an antigen presenting cell, so an APC, yeah. So it becomes antigen presenting. So you're probably thinking, why was that useful? Why? Who cares? Yeah. It's calm. All that happened here was the phagocyte attacked the pathogen, destroyed it, and it presented the antigens. Yeah. So what's that going to be used for? You'll see now in it. So now you got a specific immune response here. So you got two things. You got B cells and T cells. Yeah. B cells, they're called that because they mature in the bone marrow. Yeah. And they're involved in something called the humoral response, which is a type of specific immune response, which you'll see in a minute. T cells, yeah, they mature in the thymus gland, yeah, are involved in cell-mediated response, yeah. So you'll see that in a minute too. Thymus gland, by the way, you know that from obviously HIV, you get me, start swelling, bit of a mad thing. Alright, cool. Cell-mediated response, yeah. So we already talked about this. So we already talked about this phagocyte situation. So the phagocyte or the macrophage, they also call phagocytes that. That's more of an umbrella term for them, yeah. It ingests the pathogen, destroys the pathogen, and then it presents these antigens on the outside. You see how it presents them now? Because these are presented, the T cell can come through. T cell is just a type of like white blood cell. It will come through here, And because it sees that like lymphocyte and shit, because it sees that the antigen is being presented, it then sticks onto that. And when it sticks onto that antigen, it activates something in the T cell to start cloning. And so it produces these four things. It produces killer T cells, which are also called cytotoxic T cells, which they basically directly destroy antigen. So they just find the antigen, destroy that. That's why you start getting sore throat when you get sick here, and you need that Lemsip. It's because obviously that burn that you feel is the killer T cells killing the cells. You know what I'm saying? The helper T cells is obviously they just stimulate T and B cells. You'll see what they do in a minute. Suppressor T cells, they inhibit T and B cells because obviously when this whole thing ends here, you need the suppressor T cell to end the whole process so it doesn't keep going. And then memory T cells are very important. Memory T cells remember the antigen for future encounters. So that creates immunity, yeah. So obviously that's why you don't get sick from the same shit 2 times in a row, because you get memory T cells which are now can act quicker and they have a quicker response time. They already know the antigen, and so they can therefore form whatever they need to form to destroy it. So let's just break that down, yeah? So T cells, which are a type of lymphocyte, constantly circulate in the bloodstream and lymphatic system, yeah? When they encounter an antigen presented by an antigen presenting cell, so you need phagocytosis first, and now the phagocyte is presenting these antigens, yeah? So now the T cell can come and they can attach to it and become activated, yeah? Upon activation, the activated T cells undergo clonal expansion, resulting in the production of killer T cells, helper T cells, suppressor T cells, and memory T cells. Killer T cells destroy the pathogen directly. Helper T cells stimulate T and B cells, yeah? Suppressor T cells inhibit T and B cells, and memory T cells remember the antigen for future encounters, yeah? So write that down. That's effectively what the cell-mediated response is, yeah? So you got phagocytosis first, and then you got cell-mediated and humoral, yeah? This is cell-mediated. Alright, cool. Humoral response. So this is involving B cells, yeah? Humoral response involves B cells. Cell-mediated involves T cells, yeah? So a B cell is triggered when it finds an antigen, yeah? So again, this can be on a phagocyte, this can be on whatever. It's gonna find that antigen, yeah, which is gonna trigger it, yeah? And when it triggers it, the B cell engulfs the antigen and digests it. So it's gonna engulf that antigen there and digest that whole antigen, yeah? Now, it has these things called MHC molecules. These MHC molecules are effectively like a template. It's effectively like blue tack kind of thing, yeah? It's like blue tack stuck to a wall. What it's going to do is when the antigen gets broken down and digested, parts of that antigen are going to stick to the MHC. Yeah, it's going to stick to that blue tack, you get me? And that's what's happening here. So it basically fragments here and it sticks onto the MHC molecules. So as you see there, the antigen gets stuck there. This combination of antigen and MHC is going to attract a mature matching T cell. So these T cells, like the helper Ts and that, they're going to come through and they're going to see this situation and they're going to assist it. So it's going to basically bond onto the B cell, and what that's going to do is that's going to help stimulate clonal expansion again and cloning, yeah. Also, and the cytokines as well, secreted by the T cell, also help the B cell to multiply and mature. So when that happens, basically, the T cell is going to attach to that and it's going to secrete cytokines, yeah. These cytokines then help the B cell to mature and start cloning and multiplying. When they do that, they basically multiply and evolve into plasma cells. And these plasma cells are what actually make antibodies, yeah. So plasma cells actually produce antibodies. So then they produce antibodies and it obviously goes into the bloodstream. And obviously, we'll talk about antibodies in a minute. But this is the humoral response. So write this down. So basically, B cell is triggered when it encounters an antigen. It engulfs the antigen and digests it. The fragments bond to the MHC molecules. That causes a T cell to come and bind to it and release cytokines, which then help the B cell to mature and multiply into plasma cells. And then the plasma cells obviously produce antibodies, yeah? So there you go. So that's the immune response. Some people find it a bit difficult, but I think if you rewatch that a couple times, you note it down. It should be calm. Antibodies, yeah. So antibodies, yeah. Plasma cells produce antibodies. They're made of 4 polypeptides. You need to know that. So you've got 4 polypeptide chains here. You've got 2 light chains and 2 heavy chains. These are the heavy chains. These are light chains, and you got disulfide bridges, I believe. Um, yeah, they cause a glutination and clump to cells, which make the pathogen easy to find and destroy. So basically, what they do, all the antibodies do, is they find the pathogen. Yeah, they start binding to it. Obviously, where the antigen binds here in the variable region up there, it will obviously attach to that, and pair of them will attach together and start clumping, and that will make it bait for the phagocyte to find the pathogen. So now the phagocyte can find that, which can obviously lead to humoral response and cell-mediated response. So it just speeds things up, it makes things easier. They're essentially just markers, yeah? So they just mark where the thing is, yes? That's all they do. They just mark where the pathogen is, which makes them easier to attack, yeah? what you need to know is this is the constant region, so every antibody has the same region up to here, and then the tips of these regions, and where you see that curve, that's the actual that's the variable region, yeah. That's what the antigens are attached to. And you got one light chain there, one heavy chain there, and you got two of these, yeah. So therefore, it makes four polypeptide chains. You just need to know about the structure a little bit there. That's it. So here's an example, yeah. So the antibodies they attach to the antigens, and because they attach, it makes it bait that there's a marker there. It makes it bait that there's a bacteria there, so the phagocyte will come and destroy that, or killer T cell will come and destroy that, or anything, yeah. Antigens here obviously bind to the antigen binding site. So these are what antigens look like. They're parts on pathogens here that make them unique, and they obviously bind to the antigens there. Calm. Immunity, yeah. So obviously, natural active immunity is when you're exposed to an antigen yourself, whereas natural passive immunity is a result of crossing with like a mother's antibodies through the placenta and their breast milk. So like a mother gives to the child like through the placenta when they're pregnant and stuff, or they're through their breast milk, obviously gives them antibodies, yeah. So anything active means when your body produces it. Anything passive is when you get it. So these are both natural because they're not artificial, yeah, but one's obviously active, one's obviously passive, yeah. Active artificial immunity is acquired through vaccinations, yeah. So active artificial is through vaccination. So you haven't actually got the virus, but you artificially put the virus into yourself, or a dead part of the virus, inactive part of it, which obviously stimulated your production of antibodies in it, and the immune system to produce antibodies, yeah. Whereas passive artificial immunity is where antibodies are injected into the body. So they're already made. The antibodies haven't been made by you, that's why it's passive, but they've been injected into your body. So it's artificial, yeah. That's stuff like I think with the snake venom or whatever they talk about in it. Alright, cool. Vaccines, yeah. So it's a bit of a controversial topic, isn't it? Vaccines are dead or inactive pathogens, yeah. So they all that a vaccine is basically just a dead or inactive pathogen, yeah. So it's just a pathogen that's been made inactive or is dead. It's not gonna multiply. It's not gonna work. And on its surface, it still produces an immune response because it's still got the antigens, yeah. So it's still gonna produce an immune response, but it's not actually gonna harm you, yeah. This will lead to the production of memory B cells, which will be able to the memory cells, like the memory T cells, memory B cells, which are going to be able to trigger a rapid secondary immune response if the same pathogen was detected again. So you're it's basically like exposing yourself. I talked about earlier, if you expose yourself to a virus, you're not going to get that virus again. So all unless obviously there's mutations and things like that. But like, let's say, so all it's doing is basically just guaranteeing that you get that virus, but in a healthy way, so it's not going to attack your body or harm you. You get the virus, your body produces B cells and memory cells, and these memory cells in the future, if you do get the virus again, they'll be able to quickly act, immune response, and get rid of it. That's basically all that's happening here. The success of a vaccine depends on its cost, side effects, ease of production, and number of people who need to be vaccinated for herd immunity. Yeah, I'm gonna talk about herd immunity now. So herd immunity is when enough people are immune and the spread of the disease slows down significantly or stops altogether because there are fewer susceptible individuals for the pathogen to infect. So um, obviously, you see, if there's no herd immunity, yeah, everyone's just getting the virus, moving crazy, yeah. My man's moving like a slag, giving it to all the bandem, yeah. Now, here you got herd immunity, yeah. So obviously, that's when you got everybody's been vaccinated. So even if these men aren't vaccinated, yeah, it doesn't matter effectively because for my man to give it to them, he's got to cut through all these guys who are already vaccinated. So effectively, it gives everyone immunity at the same time. But my man who is in direct contact will obviously who's not vaccinated will obviously still get infected, yeah? That's just the way it works. But overall it will give a good it will give them immunity overall. Herd immunity is particularly beneficial for individuals who cannot be vaccinated due to medical reasons or those who have weakened immune systems. Even though they may not be immune themselves, they are indirectly protected because the disease is less likely to spread within the community. Okay, cool. So obviously got to think about the ethical concerns in it. So obviously, production and testing of vaccines may be done on animals. So obviously, there's the animal ethics side of it. The risks of the vaccine need to be balanced with the benefits as well. That's a very smart point there. The vaccine must be tested on humans as well to determine the toxicity. Yeah, and vaccines are also very expensive as well. They cost quite a lot to produce, as you can imagine. Should vaccines become compulsory or should people be able to choose? That's another ethical consideration. You might think this is fugazi, this is a useless slide. Listen, bro, when it comes in the exam, yeah, let's see how useless it is. You get me? Alright, calm. Alright, cool. Monoclonal antibodies, yeah. So monoclonal antibodies are designed to target a single antigen with a high specificity, yeah. This means that they can precisely bind to a particular target, such as viruses, bacteria, or abnormal cells, which live while leaving healthy cells unaffected, yeah. So Obviously, from GCSE, you might know how these are created. These are obviously created through injecting antigen into like a real organism such as a mouse. Obviously, their plasma cells are gonna, produce the antibodies for that. They're gonna bind to tumor cells since tumor cells replicate fast. They're like cancerous cells, and the plasma cells are gonna breed with the tumor cells to make hybridoma, and those hybridoma obviously making antibodies because they're plasma. So they got the benefits of both. They're making antibodies because the plasma cells, and they're also spreading fast because it's fused with tumor cell. Yeah. Alright, calm. And what that does here, it just makes better antibodies here, which means that the antibody can be specific now to that exact antigen, which means that it can therefore find that antigen and do whatever it needs to do, yeah? Okay, cool. Let's talk about direct therapy, yeah? So this is where monoclonal antibodies, yeah, are specific to antigens found on cancerous cells and can be used to target and destroy cells, yeah? So this is when a cell knows the antibody is exactly specific to an antigen, yeah? And therefore it's used to go and target that cell and directly destroy it, yeah? There's also indirect here, which is when you add a drug onto that monoclonal antibody. Yeah? And since that monoclonal antibody is specific to the antigen, yeah, it's gonna find where the antigen is, and since it's holding cytotoxic drugs here, it's gonna, attach to the antigen. And when it's attached, it's gonna trigger the cytotoxic drug, yeah. So it's gonna destroy that cell using the cytotoxic drug, yeah. It can also be used for diagnosis, yeah. So it can be targeted by antibodies to measure levels of the antigen in the body, yeah, as we'll see in a minute with certain tests here. Okay, cool. HIV, yeah. So you need to know about the structure of HIV. So you need to know that there's a glycoprotein, yeah. That's what's on the surface. That's for like receptor purposes, yeah. Is we got reverse transcriptase, which is an enzyme inside. It's surrounded by the capsid, and then the whole thing is surrounded by a viral envelope, yeah. And it also has its RNA inside, which is genetic information, yeah. So the core contains RNA and the reverse transcriptase enzyme, yeah. This is a retrovirus, yeah, which we'll see what it means in a minute. Envelope is an outer layer made of a membrane that taken from the host's membrane. The capsid is the outer coat. Attachment proteins allow the virus to attach onto receptors on the helper T cell. So how infection of HIV works, yeah? So the virus has these glycoproteins, yeah, which specifically bind to CD4 receptors. You need to know the name of the receptor, yeah. It's a CD4 receptor that's present on the surface of the helper T. So the helper T cells that we talked about in immunity, yeah, they got that CD4 receptor on the surface, yeah, and that's gonna attach to the glycoproteins on the virus on HIV. So when that happens, yeah, the viral and the cellular membranes they fuse together. The cell, the helper T cell, and the virus they fuse together, yeah. And this fusion allows the viral core containing the viral RNA and reverse transcriptase to enter into the host cell, yeah. So it enters into the helper T, and then the viral RNA is then reverse transcribed. So what that means here is normally in transcription we got DNA moving into RNA, yeah. So here's reverse transcription. So we're getting RNA and we're moving that into DNA. So we're forming. So the reason for that is obviously because our cells, the helper T cells, they do things in terms of DNA, obviously, whereas the virus does things in terms of RNA. So what it's doing is it's coding its own RNA, its own genetic information into DNA to then put that into the helper T cell to then replicate itself. Do you see what I'm saying? Yeah. So the viral RNA is then reverse transcribed into double-stranded DNA by the viral enzyme reverse transcriptase. That's why it's called a retrovirus, yeah, because it's able to do that. Cool. AIDS is obviously diagnosed when a person has a CD4 count, so that's a receptor count on the helper T cells, yeah, of less than 200, yeah. So that's when you have less than 200 helper T cells per cubic millimeter of blood. At that point, the HIV has spread quite far, yeah. AIDS causes extreme immunodeficiency, so it means that like you become weak, yeah. Anything can kill you, yeah. Any little disease, you know what I'm saying, flu, anything, yeah. And antiretroviral drugs, yeah, must be taken to manage it. Chronic AIDS refers to the long-term management and living with HIV infection. So once you got chronic AIDS, that means that you're with it for life. You get me? So yeah, obviously you can decrease the viral load, which means that obviously you get me. It means that you can interact with the average person in a day-to-day. If you got a low viral load and you're taking medication for that, then you're not going to infect someone. There's a low chance, even if your blood comes in contact with them. Obviously, it's a risk, though, isn't it? I don't know if you're willing to take that. Also, they claim as well. Obviously, it can only be spread through fluid transactions. You get me? So kissing and that sort of shit obviously doesn't spread it, but you can take the risk if you man want, innit? Alright, cool. Also, another thing with this too is obviously it's permanent in it. So watch out, bro, because this shit is crazy. You get me? It's a permanent thing, yeah. You got like 2 hours in it when you fill in the form. It says like within 48 hours, yeah. Obviously they can give you shit within 48 hours to help leave it if it hasn't moved to the bloodstream already. And yeah, you can also take if your job involves like things to do with blood, like taking blood, shit like that. You can also take PrEP, which is like, medication that you take beforehand so that even if you come in contact with HIV, it's not gonna do shit. So yeah. Okay, cool. Direct ELISA test or ELISA test, yeah. So monoclonal antibodies are, so this is basically a test to test for different shit, so for different antigens, yeah. So you can use it to test for many different things like coronavirus, like pregnancy, like whatever. This is a form of direct test. So obviously this is a direct one. I'll talk about the indirect one in a minute. So the direct one is this is like a, yeah, is basically monoclonal antibodies, yeah. They're attached, they're immobilized, so they don't move. They're just attached to the bottom of a plate, yeah. And antigen molecules present in the sample bind specifically to the immobilized antigen on test plate. So what I mean here is, let's say you got trying to test for HIV, yeah. What happens here is the monoclonal antibodies, yeah, that attach to the plate, yeah. And these monoclonal antibodies are obviously complementary to HIV antigens, yeah. So it's on the plate, and when you add the blood, yeah, the blood binds specifically to the antibodies, yeah, on the plate. And then a secondary antibody will be added, which has an enzyme on it, and that has to bind to the antigen antibody complex that's now formed. So if that complex has not formed, this secondary antibody is not going to bind. And after it binds, you wash it obviously to remove any unbound antibody, yeah. After it binds, if it binds, it will catalyze a reaction, and you add substrate into the whole mixture, and that substrate is obviously going to be catalyzed by the enzyme, and that's going to result in a color change, which is going to be detectable. So just a little recap there. Basically, what's going to happen, I'll tell you a positive test. So a positive test is the monoclonal antibodies are immobilized on the bottom of the plate. You add blood into it. That blood has HIV, so it attaches to those antigens on the bottom of the plate. Then you add the enzyme antibody inside. The enzyme antibody attaches to it since it's present, yeah, since it did form that complex. And then once it attaches to it, you add substrate, and that substrate is going to be catalyzed by the enzyme that's attached to the antibody, yeah, and that's going to cause a color change. Do you see? If it wasn't present, so let's talk about if it wasn't present now. If it wasn't present, you're going to add that same, so the antibodies are still there on the bottom of the plate. You add your blood doesn't have the antigens present, so it's not going to stick to it. Therefore, the complex is not made. So therefore, when the secondary antibody is added, it's not going to bind to it. So therefore, when you add substrate, it's not going to do shit because it's not binded anyway. Do you see what I'm saying, yeah? So that's how it works. Always, yeah, when you write it in the mark scheme, yeah, when you're writing it in the question, always mention that you have to wash after adding the test samples. After you add the blood, yeah, you obviously wash it to remove any unbound test sample, any unbound antigens, yeah. And also you wash as well after adding the antibody. So when you add that second antibody, yeah, you need to also wash it as well to remove any unbound antibody, yeah. There's actually a little like conspiracy thing as well surrounding this ELISA thing, yeah. I don't know if you man know that video of that girl that went missing or whatever in the elevator, doing some madness in the elevator. You get me? Obviously her name was like Eliza as well, and like when she went missing, this test got developed, fam, and in the area where they were in it to cure some disease in the area. And like some people claim in it that they used her as like, you know what I'm saying, testing or something in it, because her name and the name of the test are the same, like the full name is the same in it. So a bit of a mad thing there. Pregnancy test, yeah, a lateral flow. Monoclonal antibodies specific to HCG, yeah, a hormone made during pregnancy, yeah, are immobilized on the test line of the strip. So you get a test line on the strip, yeah. So this is like corona test, lateral flow, yeah. The reason it's called lateral flow is because it's moving laterally, yeah. So monoclonal antibodies, yeah, they're specific to the HCG hormone, yeah. That's the hormone that you make during pregnancy, yeah. So they're immobilized on the test line. So these monoclonal antibodies, they're just on the test line of the strip on the bottom. They're just stuck there. Then the person adds their urine into the sample pad of the test, yeah. If HCG is inside the urine, it's going to bind to the mobile antibodies, not the immobilized ones, the mobile ones. It's going to bind to the mobile antibodies that are already inside the strip. And these mobile antibodies, yeah, they're attached to colored particles or enzyme kind of situation, yeah. And so what happens here, this HCG antibody color particle complex is going to move along the strip. So the HCG binds to the mobile antibody, which moves along the strip, and then once it gets to a certain point, so when it reaches the test line, the immobilized anti-HCG antibodies, they capture the complex. So there was the immobilized ones before, yeah. We added the HCG, your urine, yeah, which has HCG. That HCG attaches to the mobile ones. The mobile ones move it across up to where the immobilized ones are. The immobilized ones capture it, yeah, and because they capture it, a visible colored line appears in the test line, showing a positive result. Also, the standard urine is also going to continue moving along to a control line, which is always going to be show a colored line, yeah, to show that the test works properly, yeah. So whatever you add, the urine is going to look for the antigen, yeah, regardless, even if you don't have any HCG, and it's going to show that control line, like using with coronavirus, yeah. If you take the test, there's always one line that appears, and the second line is the test line, yeah. So just a little recap there. So what happened there is that there's already antibody specific to HCG that are immobilized on the test line. The person adds a urine sample. The urine has HCG, so therefore the HCG is going to bind to the mobile antibodies that are already inside the sample, and they're going to move it across to where the immobilized ones are. The immobilized ones are going to attach to it from the other side, so it creates like a sandwich effect. They call it as well ELISA sandwich test here. And then obviously because of that, that's going to form, because you got a antibody from one end, and it has a colored particle on it, therefore it's going to form a line. It's going to form a colored line, which shows a positive result here. And if there was no positive result, there's also obviously a control result, which is the urine continues moving to the control line, which is always going to show a color line to confirm the test worked properly here. So yeah, so that's the end of the whole situation. That's the end of the whole topic here. Alright, pattern.